This conference will now be recorded.
Hello, everyone. I think we still have
Oh,
a few people jumping on, but I'll go ahead and get us started with a couple of housekeeping items. If you could please keep yourself muted and your video turned off unless you're one of our Public Oversight Committee members or guest speakers. That would be a big help to us. It helps with our recording and helps make sure all the Public Oversight Committee members can see one another. I'm going to start by sharing my screen with tonight's agenda before I kick it over to our chair. Okay, so just a reminder for anyone who hasn't joined us before, this is a meeting of the 1921 Race Massacre Graves Investigation Public Oversight Committee. The Public Oversight Committee was established to ensure transparency and community engagement throughout the 1921 Graves investigation. The committee serves in an advisory capacity to the city during key decisions throughout the investigation. The Oversight Committee is comprised of descendants of the Tulsa Race Massacre and leaders in Tulsa's African American community. By the end of our time together this evening, we will receive information and have an opportunity to ask questions about how DNA analysis and identification might support our efforts. And so with that, I am going to stop sharing my screen and turn us over to our chair, Kevin Ross, for our welcome.
hello, everyone. Can you hear me?
Yes, you sound great.
Okay. I'd like to welcome not only everybody from near and far, but also I'd like to have a shout out to the 4th and 6th grade students at McKinley Elementary School who are at this time studying about the massacre of 1921 and also watching everything that we're doing here in Tulsa in reference to our mass graves investigation. I also like to state that in our hearts of hearts, the world is watching us and our children are watching us as well. It's a beautiful thing right now when everybody's learning at the same time. And I think we're at the right place at the right time on our watch to make things happen. I'm excited about what we get ready to venture, something that's been long awaited for such a long time. And I'm glad and welcome everyone near and far and around the globe to Tulsa as we begin our first steps towards making good history. And I thank you.
Thank you, Kevin, for that welcome and introduction. I don't believe we've been joined by Reverend Turner yet. Kevin, would you mind leading our moment of silence this evening?
At this time, everyone, can we please have a moment of silence before we begin on our journey? And I thank you.
Thank you, Kevin. So just as a refresher, at a prior Public Oversight Committee meeting, Dr. Stubblefield gave a short overview, provided a little bit of background information about DNA analysis. And one thing that the Public Oversight Committee requested was an opportunity to speak with someone who really had this as their primary area of expertise. And so through referrals, we were able to be connected with Dr. Robert Allen, who is joining us on video and has put together a much more in-depth presentation for the Public Oversight Committee this evening. And so I think he's going to introduce a little bit of his background as part of his presentation. So if you are ready, we will turn it over to you, sir, and you can share your screen, and I'll let you say a few words of introduction.
Okay, thank you, Amy. Thank you all for having me, giving me the opportunity to talk to you this afternoon and perhaps be part of this process. Let me get my screen shared here. It's not letting me share, Amy. I think you have to make me a presenter or something. I'm clicking on the link, but it's not getting me anywhere.
Let's see, we'll re-enable you. Okay, we have you set as a presenter.
Let's see if I can.
I can also share your slides if needed. Every platform has its own weird.
Yeah. Okay, I guess you're going to have to do it because it just keeps saying that it needs to use accessibility features, and when I do that, nothing. Okay. You got me up?
I've got you up, and you just tell me when you're ready to advance.
Okay, so let's start over. Thanks for the opportunity to talk to you. As Amy said, my name is Robert Allen. I'm a professor of forensic sciences at Oklahoma State University Center for Health Sciences, which you may know more, heard about more as the College of Osteopathic Medicine, close to downtown, 17th and Southwest. And I'm going to discuss with you DNA analysis as it applies to human identification and how the status of the field nowadays is such that it may be very useful for identifying the remains at Oak Lawn Cemetery, and I'm going to propose a possible strategy going forward to investigate those remains and try and identify them. So if you can go to the next slide, please. So first, a little bit about me. I'm a Tulsa native. Grew up here my whole life except for 5 years that I spent with my parents in Venezuela when my father worked for an oil company. Educated in Tulsa Public Schools and the University of Tulsa, where I received a bachelor's degree, then moved on to take a PhD from Purdue University in West Lafayette, Indiana, and following receipt of my degree, moved to Southern California, and did 3.5 years of postgraduate training in molecular biology and molecular genetics. My first real job was with the American Red Cross Blood Services in St. Louis, Missouri, which is where I got interested in DNA-based identification in the mid-80s. So I've been at it for a very long time and have been in the very fortunate position to watch this technology in this field evolve to the point it exists today. In St. Louis was where I began to apply my knowledge to for the St. Louis County Courts and the St. Louis County Police Department, providing parentage testing using DNA-based methods as forensic DNA testing in criminal investigations for the St. Louis County. Moved back to Tulsa in 1992 when my kids were small because I wanted them to grow up with knowing extended family on a regular basis, and all of my family was still here in Tulsa. Joined the scientific staff at Children's Medical Center, and for those of you who have been in Tulsa for a long time, you'll remember Children's Medical Center as the place that always decorated extensively for Christmas out on Skelly Bypass, just east of Yale. And I worked with the H.A. Chapman Institute of Medical Genetics there. When I moved back to Tulsa, I was doing some of the early DNA testing for the Tulsa Police Department before they had that capability in their own laboratory. Moved to the School of Forensic Sciences at OSU CHS in 2001, as the School of Forensic Sciences was just getting started, and was charged as a member of the faculty with developing a training program for forensic DNA analysts. And since joining the faculty at OSU CHS, we have been involved for quite some time, identifying remains through kinship testing, a challenge that is sort of in the same ballpark as the project that we're talking about today. If I can have the next slide, please. So first, we'll get a little primer on the structure of DNA because it's very important for the discussions that follow. When you hear people talk about DNA, invariably you'll hear it referred to as a double helix. The best way I can describe the double helix is taking an extension ladder and sort of twisting it on its center axis, where the rungs of the ladder are composed of interactions between simple chemical compounds known as nucleotides that interact with one another in a highly specific way, something known as base pairing, that is generally thought of as holding the helix together. So the sequence of these simple chemicals specifies the set of instructions that constitutes our genetic blueprint that defines who and what we are. And by and large, of the 3.2 billion nucleotides that are linked together in chains that compose the chromosomes that we all have, the vast majority of that nucleotide sequence is exactly the same between you and me and every other human on the planet. There's less than 1% of the nucleotide sequence that varies among the human population, but it is this really tiny percentage of our genome that is the focus of our investigations when we're doing testing of kinship, criminal investigations involving DNA. We're looking at a really small proportion of our genome that varies from one person to the next, but these variances are preserved within family groups. If I can have the next slide, please. So there's really two types of variations in human genomic DNA that are relevant to our discussion today. One that has been around for quite some time and is used extensively in the fields of parentage and kinship testing, as well as DNA testing, are genetic markers that consist of tandem repeats, also known as STRs. And these are small repetitions of short sequences of nuclei, usually 4 to 5 in length, that are tandemly repeated over and over and over again along a length of chromosome. And since we have capabilities of precisely measuring the length of pieces of DNA, we can distinguish a piece of DNA that has 7 of these repeats from a different piece of DNA that has only 4. Or likewise, we can match 7 repeats in DNA obtained from a buccal swab or a blood sample from a suspect in a criminal investigation and match it against the same 7 repeats found in recovered from a crime scene. So STR typing is probably the most widespread form of human identification testing practice on the planet. Another major category, and one that's more relevant to our discussions today, are genetic markers consisting of single nucleotide substitutions. In other words, specific positions within a chromosome at which the nucleotide that occupies that position may vary among different individuals. So in our example, our cartoon here at the bottom of this slide, you see we have chromosome from person A and a chromosome from person B. And by and large, the sequence of nucleotides in all those positions are identical among these two individuals. However, if you go down to the next to the last on the right and the last on the right, you notice that the nucleotides are different in those positions. You still have C pairing with G in one chromosome, but in the chromosome from the other person, you have an A-T pair occupying that position. So this is only a cartoon piece of chromosome 24 nucleotides long, but if you extend this out to the 3.2 billion nucleotides that constitutes the human genome, you can see that you can have many, many opportunities for variations of particular nucleotides at particular positions within our chromosomal DNA. And it is single nucleotide substances, for example, that are often associated with genetic diseases like hemophilia or sickle cell anemia. It's also single nucleotide polymorphisms that have been traced to particular ethnic backgrounds and particular physical traits like eye color, hair color, etc. And it is also single nucleotide polymorphisms, or SNPs, as they're abbreviated, in our chromosomal DNA that are very useful for uncovering distant familial relationships. And both of these types of polymorphisms are widely, exist widely in nature in virtually all species. So if I could have the next slide, please. So everybody's DNA is different, but members of the same family are going to share portions of their DNA. In other words, pieces of their chromosomes, and this is going to become a little clearer in a moment, depending upon how closely related they are. So if I could have the next slide. So as I said, SNPs are abundant in human DNA. In that 0.6% of the DNA that varies among humans, there's about 4 to 5 billion SNPs that are scattered over all 23 pairs of chromosomes, 22 pairs of non-sex chromosome and a pair of sex chromosomes. And in addition, SNPs exist in a different form of DNA that exists within every cell of our body, known as mitochondrial DNA. Mitochondria are sort of the little energy factories that keep us going during the day, providing energy that we need to live. And in our cartoon here, you see we have two chromosomes that may exist in an individual. One's painted black, one's painted red because of the sequence of SNPs that exists along the length of these chromosomes. Every vertical hash represents a within the chromosome. In our cartoon here, where a single nucleotide polymorphism exists. And in some cases, they're exactly the same in the two chromosomes, but in other positions, you can see that they differ. Well, as reproductive cells, sperm and egg, are produced in someone who's about to be a parent, there is a shuffling of the portions of these chromosomes that occurs as a result of a phenomenon known as recombination or crossover events. And so the integrity of chromosomes gets shuffled as gametes, or a sperm and egg, are produced in an individual. And so as a family traces back to a common set of parents, the amount of family DNA that will be shared between two individuals is going to increase. Let me clarify it by going to the next slide. Maybe that'll make a little clearer sense to you. I didn't really state that very clearly. So if we have these two chromosomes in a parent to be, painted black, painted white to show the sequence of SNPs along the length of chromosome, with a recombination event, we sort of break these two chromosomes in pieces and rejoin them, and now we have a thing that sort of looks like a hybrid. And so the chromosome that gets passed to an offspring conceived with one of these chromosomes, even though the same DNA is present in that child, the chromosome's not going to look like it does in the parent that it came from. If I can have the next slide. So as we go down through generations, what you see is that the chromosomes in offspring look less and less like their ancestors. who gave rise to them as you go back up through the pedigree. However, even in the bottom cartoon here, even in the child, you can still see remnants of that original grandfather's chromosome and the original grandmother's chromosome present in their chromosomal makeup. So as recombination occurs throughout the generations, the amount of family DNA that gets shared becomes less and less as you look at descendants. But there's still a significant amount of sharing that takes place, and you can detect that sharing based upon the sequence of SNPs that exist within those chromosomal fragments. If I can have the next slide, please. So here we sort of see this in a pedigree form, where if you map DNA against DNA of your relatives within your group, whether they be close relatives like your grandparents or great-grandparents, or distant relatives like a third cousin once removed or a third cousin twice removed, notice that circled in each of these boxes is something that says average 1760 cm. Well, cm refers to a centimorgan, roughly translates to about 1 million nucleotides of sequence. So you would be expected to share with your grandparents about 1,760 centimorgans of chromosomal pieces. All right, that's the total length of all the sharing amongst all 23 pairs of chromosomes between you and your grandparents. If you compare that with the amount of sharing with the third cousin once removed, bottom right, you see that there's only about 76 centimorgans of DNA that has been shared, reflecting the additional generations that have existed leading to that third cousin once removed. And as I said, the more you are within the pedigree, the smaller the pieces of DNA that will be shared with your relatives. However, the amount of sharing, even between a third cousin twice removed, is going to be significantly higher than the sharing that two random individuals who are unrelated will exhibit when their SNP genotypes are compared. If I can have the next slide, please. So the first real public application of this kind of technology was with companies like 23andMe and Ancestry.com. I'm sure everybody's been exposed to ads for Ancestry.com on any number, in association with watching any number of television shows, or you see it in the newspapers. And although these sorts of applications got their start, giving clients a prediction of where they originated on the planet, especially important here in the United States, because just about all of us are from somewhere else. And so it's nice to know, or it's interesting to know, it's coffee table talk, if you will, to know that you're evolved from or descended from British Isles or Sub-Saharan Africa or Asia, whatever it may be. But the state of the technology as it exists now is such that people are using it for genealogical studies to expand their knowledge about their own family trees. And that's really the piece of this that I think is key for the challenges we face in this particular project. Next slide, please. In fact, I got this ad yesterday as an email from Ancestry.com advertising a Mother's Day gift for 59 bucks. You can have this done. So it's not terribly expensive. And the fact that it's not terribly expensive has prompted a very large number of clients to subscribe to Ancestry.com and to submit their samples. In fact, I've submitted my own sample. If I could have the next slide, please. And there were 2 notices that I've received. The first one involving a guy named William Hill, who Ancestry.com says is a third cousin with a very high confidence. And you can't really see the picture here, but Mr. Hill is holding in his arms a 4-foot alligator, and he is supposedly a third cousin. 2 weeks ago, I got another notice from Ancestry.com that they had identified somebody else they believed to be a third or fourth cousin who goes by the username of I See a Rat. And his posting with Ancestry includes a family tree of his. So you can see how this would reel to the hobby genealogist, using these DNA-based matches of chromosome segments to identify extended members of their family tree. But either of these guys, they come out of the blue. Frankly, based upon what I know about them from just looking at the notices for tree, these may be branches of my family tree that come from the shallow end of the gene pool. I don't know, I'm just saying. All right, if I could have the next slide, please. So the technology has demonstrated itself to be very powerful, even in my own personal life, and has shown success in detecting distant family relationships. And our strategy, I believe, should begin with an Ancestry.com kind of process. For contract testing, it's not tremendously even at retail, and I believe some companies might be very interested in this project and perhaps even be willing to donate this service to advance our cause. If I could have the next slide, please. So what could test results give us to advance our cause? Well, first, I think we could get a prediction of ethnicity on all these remains in these graves. We could determine if the predominant ethnicity is African American. We could get information about possible family relationships among the interred. It is possible that brothers or parents and adult children or cousins, families may have been victims of the race massacre and may have been interred together in the graves that are uncovered. And I believe this would be very useful information for tracing the identity of those folks. We could also establish portfolio links to folks in the database who have submitted samples in the past. As I said, there's been a really large segment of the population that has subscribed to Ancestry.com already. And so presumably their SNP genotypes are available for comparison against those that were produced from those interred in these graves. I mentioned people who believe they are descendants of race massacre victims could submit samples currently to get their data in these databases that will be searched. If I could have the next slide. So what are the questions? What sample are going to be used for DNA recovery? Skeletons consist of bones, and can we extract useful DNA from the available samples? We have some experience working with human remains, as I indicated in my introductory comments. We've worked with bones, we've worked with samples of both types that were exposed to the elements for 20 to 30 years. Now, that's not 100 years. And so we're in an uncharted area here, at least in my own experience. But all we can do is try and assess the quantity and quality of DNA we recover from those remains we analyze. We need to know how much DNA we recover from the bones we work on. We have with which to quantify both nuclear and mitochondrial DNA. We've been doing it for years. They're very sensitive, so even if we're low quantities of DNA, I think we'll be able to get reasonably accurate quantity estimates. And the methods we use are the same as those used in crime laboratories all over the world. A very big part of our analysis, or a very big question we need to ask, is what the integrity of DNA we recover is likely to be. DNA degrades with time, especially in a wet environment, and it just continuously fragments into smaller and smaller pieces. And at a certain point, the pieces become too small to be useful for SNP genotyping. We also have methods with which to assess the integrity of DNA that we recover to estimate how broken down it's likely to be. And knowing answers to these questions is going to allow us to make some informed decisions on how best to proceed with developing a testing strategy. If I could have the next. So here's the workflow that I'm proposing. I'm proposing that we work systematically through the remains of the bird, extracting DNA from the tooth and from a weight-bearing bone, usually or the femur, a leg bone, to assess quantity characteristics of the DNA recovered from each skeleton. It may very well be that individual skeletons are going to yield DNA in different quantities or different qualities, kind of depending upon where they are in the grave and what sort of environmental conditions they're exposed to. Some of the coffins may have collapsed, may have allowed more moisture in. Some coffins may be in areas that are wetter than others in the grave. And so one size may not fit all in terms of all DNA samples and remains can be quantified and subjected to integrity analysis here at the OSU Human ID Laboratory. I've mentioned our possible involvement in this project to OSU administrators, and they seem to be totally on board with providing services in support of this in this project. And I would also propose that simultaneous with workflow step number 1 would be to make an open call to the community for sample donations so that those SNP genotypes are assured in the database. All DNA quality data will direct our selection of a genotyping service. Could be Ancestry.com, 23andMe, and there's others. And of course, considerations for choosing a company are going to be cost, but almost more importantly are the searching capabilities that they offer, what's going to be reported back to us. And then also, although I don't believe there is accreditation, formal accreditation available for companies like Ancestry or 23andMe, there is a fairly well-established set of quality assurance practices that we want to make sure are in place with any company we contract with. And we also want guarantees of data privacy and data security. Next slide, please. If we do identify some potential kinship relationships, there is some additional selected kinds of DNA testing that can be performed if the DNA recovered is sufficiently intact. If you remember, one of the types of genetic markers I mentioned early on was STR type markers. Well, these guys exist on the Y chromosome, which is inherited patrilinearly within extended families. So if you're a male, your Y chromosome is going to match the Y chromosome of an uncle or a grandfather or a male cousin who is the son of your father's brother. And so if we identify potential relationships using SNPs, we can strengthen our confidence of those relationships if they are individuals and they share common YSTR marker haplotypes. All right. The same applies to testing mitochondrial DNA analysis, mitochondrial DNA, in the sense that it is inherited matrilinearly. So it's inherited within the female family line. And so if you're a male, you're going to share mitochondrial DNA with your mother, with her sister, with her mother, with a female cousin. You see where I'm going with this. My laboratory performed Y chromosome analysis and would be in a position to provide that service should it be required. But mitochondrial DNA is going to have to be contracted. We don't do that. There happens to be a laboratory in Oklahoma City that does do mitochondrial DNA, so we don't have to look too far outside the Tulsa city limits to find a source of that service. And I think the project's going to require the services of a genealogist, especially one used to combining the DNA record with that constructed from traditional sources of public records, etc. And if I can have the next slide, please. That's all I got to say on the subject. Thanks for your attention. If there's any questions, I'll do my best to try and answer them.
Thank you very much. I'm going to stop sharing our screen. So if Public Oversight Committee members want to unmute and show their cameras, we can have this be more of a discussion. Are there any questions?
Yeah, yes, I have a question. I know, so this process, how long does it take to get the needed results in order to pull off what you're saying, to get the results where we are able to go back in time and basically claim some kinship to folks?
Well, it's going to depend upon two things. It's going to depend upon whether or not relatives exist in the database. The DNA profiles obtained from the remains are compared against. These databases expand continuously. I don't know how many folks are in Ancestry.com's database, but I suspect it numbers in the millions now. And if you think about a third cousin twice removed, that's a pretty distant family relationship. And even though you know members of your family quite well, just like me, I didn't know about Mr. Miller or I see a rat. I didn't know they existed, but they at one point or another submitted samples to Ancestry.com and had the same service provided them that was provided to me. And Ancestry made that comparison and made that match. So the length of time it takes is going to depend upon, is the database of sufficient size to find a match? In terms of actually producing the DNA profiles from the bones, once we have DNA and have an understanding of how much we've got and its state of integrity, I think the Ancestry.com typing process doesn't take very long at all, maybe a week or so. In terms of extraction, in terms of the quality analysis that I'm talking about for each set of remains, that's probably just a matter of 3 or 4 days. So the front end of the actual processing can be done rather quickly. Getting, if members of the community in Tulsa or in the state know that they are descendants or have good evidence that they are descendants of victims of the massacre, they can submit their samples and, as I said, ensure that they are in the database for matching against profiles produced from the remains that are in the grave. Does that answer your question?
Yes, and also I've over the years seen numerous ads about Ancestry.com, and people who did buy those packets where they were able to find out what part of the country your ancestors came from, or what part of the world that they really based out of, and really get into genetics. And I'm just trying to figure out how would that work with a community of people. Most of, like, my family came upon the Trail of Tears, who were brought as slaves to what we know today as Oklahoma, whose DNA probably stretched over numerous cultures. How reliable is it? I mean, do you get your money's worth, or they just got a package and they just slide it in and got to get your money? How does that really work, and especially for the process that we're talking about now?
Well, I can tell you that 10 years ago, if you saw an ancestry report, it was filled with all kinds of disclaimers and waffle language and everything else, that if you really looked at the fine print, you really wondered about the accuracy of the results you were getting. Nowadays, that's not really the case. Nowadays, criminal investigations of cold cases are being directed by these types of tests, defining the ethnicity of the unknown suspect who committed that crime, who's never been identified. So if law enforcement knows they're looking for a Hispanic man, they trust those results sufficiently to no longer investigate Caucasians or African Americans or Asian individuals, whereas without that information, all of that effort would have been expended investigating people who don't match the ethnicity characteristics of the perpetrator of the crime. So I think the confidence level is high enough that if you were to have this done, your results would come back and show that you have mixtures of DNA from Native American, Black individuals, etc. And maybe you can't really, you wouldn't bet a month's paycheck on splitting hair, 5% Scandinavian, 4% German, 22% Jamaican, etc. But in terms of large ethnic groups, Black, white, Hispanic, Asian, etc., you could take that pretty well to the bank. And we don't know at this point, as I understand it, that the remains that are interred in that grave are African American, Caucasian, Asian, a mixture of all. We don't know much about them at all, and that's why I think that information is important to advance our understanding of what we're working with here. And it's just a byproduct of getting the test done anyway.
And I asked this question to Dr. Phoebe on a number of occasions. One, the condition of the remains, and would determine the results that can be brought out for the DNA extractions. Would there be any kind of problems if the body has been embalmed or not? Would that be a contamination of the DNA? Anyway, does that interfere with the DNA extractions?
Well, if you're asking me that question, embalming, yes, does present a problem, but that's why we deal with the long bones of the leg, the weight-bearing bones of the leg. They're weight-bearing, and so the cortex, the thickness of the bone, is at its maximum there. And it's a really dense, it's almost like a rock material. And embedded in the matrix of the bone are the cells that are responsible for keeping it alive. So that tissue, if you could call bone a tissue, is not well vascularized. So when they run those preservatives through the body during the embalming process, the cells embedded in the bone don't get exposed to it immediately. And only with time does it have the possibility to seep through the matrix of the bone and ultimately affect the integrity of DNA in those embedded cells. I can't predict in 100-year-old bones what the outcome is likely to be. All we can do is give it a shot and see what we get.
Thank you. Thank you much.
So Dr. Allett, I just wanted to ask a clarifying question. The proposed workflow that you talked us through is really sort of sounds like a two-phase process. One phase would happen immediately in the field this summer, collecting samples and determining the integrity of those samples. And then sort of the second phase would be, we'd probably have to do a competitive process as a government entity, but then selecting a lab partner that would help us with the identification part. Is that an accurate understanding?
Yep, that's how I envision it. We go to a company or a handful of companies with a portfolio of biochemical characteristics of DNA we've recovered from each of the skeletons that are unearthed. And so we know going in exactly what the quality characteristics of the sample recovered from each skeleton is. And thus, I think we're in a better bargaining position to say, look, this is a really high quality sample. You're not going to have to expend extraordinary efforts to get the DNA results we want. So we want our brother-in-law deal on this one. This one over here may be a little more difficult. I don't really think that those discussions are going to be nearly as intense as I'm intimating, because I think companies are going to jump at this because of the notoriety of the project.
That's very helpful. And just to reiterate, for the Public Oversight Committee, the portion of this work that's being offered by OSU Center for Health Sciences and would be funded by them would just be DNA sample collection and determining the integrity of those samples. Is that correct? Okay.
Well, in the event that there's some of the specialized testing needed, in the last couple of slides, I talked about testing STR markers of the chromosome, be in a position to make that available as a donation as well.
Thank you. I know we have a few other Public Oversight Committee members on that we haven't heard from. Are there any other questions from the Public Oversight Committee?
Either I did a really good job or I've got everybody so confused they don't even know what a question to ask.
Well, thank you for your time. Thank you for providing this deeper dive. I hope the Public Oversight Committee members who requested more information about what a DNA analytical plan could look like, I hope that this answered that. As a reminder, the video from tonight's meeting, as well as the slides, will be posted on the city website at cityoftulsa.org/1921 Graves. Kevin, were you going to add something?
Just one thing. Thank you, sir, for giving us a better understanding and illustration. Understand that it's not like it happens on television, nor on those fiction novels, but give us more clarity to what to expect and what we're up against. And to that, we don't know until we actually go get them. So thank you for your time and explanation.
That's been my pleasure. Hopefully our paths will cross again down the road.
All right. Thank you very much. We're going to move to the next item on tonight's agenda, which is our questions from the public. You can enter those in the chat, and we will allow some time to go through those. There have been quite a few questions about how people can get involved, and we're encouraging anyone who's interested to register with the City of Tulsa as a vendor. As a governmental entity, we would go through a competitive process to select a partner for this work. All right, question from Chris Polanski. Is there a timeline for when this work will resume? I believe Dr. Stackelbeck saying last meeting possibly starting on June 1st, and that's correct. We're working diligently towards being ready to start on June 1st. A slight update, our funeral director has submitted the legal paperwork to the Oklahoma State Department of Health, and we're currently working with our funeral director through that process. Let's see. The press release indicated we would be talking about logistics this summer. Are we going to talk about that? Really, the purpose of tonight was to focus on the DNA analysis. That was the topic that the Public Oversight Committee had asked it for a deeper dive on. And so we asked Dr. Allen to join and provide information on that tonight. The key point is we are still planning to start work on June 1st. We received 9 responses to our request for proposals for additional archaeological services. And so our project principals will be reviewing those responses in the coming weeks and making a selection to bring some additional manpower on board to help with this project. Let's see, sorry. Every time we get a question, my chat freezes. It says, will there be an opportunity for the Public Oversight Committee to hear about the genetic genealogy process, specifically from extraction to identification? Angela, maybe we should talk offline about what additional information, in addition to what was shared tonight, you're referencing, and maybe we could identify a time to do that. We have, at the request of the Public Oversight Committee, dedicated our May meeting to a discussion about the potential for criminal prosecution to arise from this work. And we will be having Judge William Kello joining us in May to talk more about the legal aspects and implications of the project then. There was also a question in the chat on, is there an update on the Rolling Oaks GPR? Is that still expected to start at the first week of June? Yes, there is an update. We've been told by the Oklahoma Archaeological Survey that there's a commitment for that work to take place June 14th through 16th. We actually don't want that work to start the first week because the Oklahoma Archaeological Survey will be helping with the total station mapping to set up the work at Oaklawn, and we're going to need them on site at Oaklawn the first week. But once we get underway at Oaklawn, they'll be able to do that work at Rolling Oaks the 14th through the 16th. Can we say yet where the new archaeologists are coming from? No, we cannot. We just received responses today. We just minutes before this meeting had their responses available, and so we're just at the very beginning of reviewing those, but we'll go through the city's formal procurement process for that, and we will be announcing the selected vendor of that process publicly. Angela, I will put my email, I will send you my email so you can reach out. That would be very helpful. At this point, not seeing any additional questions in the chat, I'm going to move on to new business from the Public Oversight Committee. Is there any new business?
I just had one question. You might have said it the last meeting, and I didn't hear it tonight. You might have said I must have missed it. On the judge's order, has that been approved, and who was the judge? And I know pretty much it must have been approved because that's we got us a date for the actual excavation. What is the process? What was that process like, and what's the update on that for clarification?
That's a great question. So I know we actually have our funeral directors on this evening, and they may want to add comments here as well, though they're not required. So they have submitted their paperwork to the Oklahoma State Department of Health, and we provided a letter documenting that the city is requesting our funeral director to assist us with this project. We outlined the scope of the project, and we also cited the city's authority under the next of kin statute to do this work. And so right now we are waiting for the Oklahoma State Department of Health to finish their review of that. We do have a draft of a petition for a court order that if the Oklahoma State Department of Health tells us that the statutory authority is not enough, that they want a court order, then we will be reaching out to meet with the presiding judge at Tulsa County and basically present that petition for a court order to the court. So we're still on the administrative part of that process with the Oklahoma State Department of Health, but we do have that petition ready to go should they require it.
So no judge has been appointed yet at this time.
That's correct. That's correct. And we would basically petition the court for an order, and the document that to do that process has been prepared. We received some assistance from the Tulsa County District Attorney's Office with that process. And so if the Oklahoma State Department of Health tells us it's required, we are ready to roll. Is there any other new business from the Public Oversight Committee? Okay, well, with that, if there's nothing else to be added, we will go ahead and conclude for this evening. Thank you all very much for your time and your attention and your thoughtful questions. As a reminder, all of the information from tonight's meeting, our meeting video, and the slides from this evening will be available on our website, which is www.cityoftulsa.org/1921 Thank you.
Bye-bye.